Remember the first time you set foot at UPIS? This is the gate that welcomes you. Before you enter the UPIS building, you need to pass through a gate in a fence that surrounds the school grounds. This fence prevents people who should not be there from entering, and the gate allows faculty, students, and staff to enter. Prokaryotic cells and eukaryotic cells have a similar structure that maintains control of their internal environments. Recall from Unit 2 that the process of maintaining homeostasis or balance in an organism's internal environment is one of the key characteristics of a living organism. Homeostasis is essential to the survival of a cell. One of the structures that is primarily responsible for homeostasis is the plasma membrane. The plasma membrane is a thin, flexible boundary between a cell and its environment that allows nutrients into the cell and allows waste and other products to leave the cell. All prokaryotic cells and eukaryotic cells have a plasma membrane to separate them from the watery environments in which they exist. Now let us watch this short video describing a plasma membrane.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
From the video, a cell's plasma membrane defines the cell, outlines its borders, and determines the nature of its interaction with its environment. A plasma membrane's principal components are lipids, or phospholipids and cholesterol, your proteins, your carbohydrate attached to some of the lipids, and proteins. The plasma membrane is composed of a phospholipid bilayer in which 2 layers of phospholipids are arranged tail to tail. The 2 layers of phospholipid molecules make a sandwich with the fatty acid tails forming the interior of the plasma membrane and the phospholipid heads facing the watery environments found inside and outside the cell. Each phospholipid is diagrammed as a head with 2 tails. The phosphate group in each phospholipid makes the head polar. The polar head is attracted to water because water is also polar. The head is considered as hydrophilic or water-loving areas of these molecules because they are in contact with the aqueous fluid both inside and outside the cell. On the other hand, the tails are considered hydrophobic or water-hating molecules because they interact with other non-polar molecules on both the cell's exterior and interior. Thus, the membrane surfaces that face the cell's interior and exterior are hydrophilic. In contrast, the cell membrane's interior is hydrophobic and will not interact with water. Therefore, phospholipids form an excellent 2-layer cell membrane that separates fluid within the cell from the fluid outside the cell. Moving with and among the phospholipids in the plasma membrane are proteins, cholesterol, and carbohydrates. Proteins at the inner surface are called integral proteins. When found on the outer surface of the plasma membrane, they're called peripheral proteins. Some integral proteins have a hydrophilic channel to their center that allows passage of hydrophilic substances. Peripheral proteins are on the membrane's exterior and interior surfaces, attached either to integral proteins or to phospholipids. Peripheral proteins, along with integral proteins, may serve as enzymes, as structural attachments for the cytoskeleton's fibers, or as parts of the cell's recognition sites. The cell recognizes its own proteins and attacks foreign proteins associated with invasive pathogens. Carbohydrates are the third major plasma membrane component. They are always on the cell's exterior surface and are bound either to proteins forming glycoproteins or to lipids forming glycolipids. Along with peripheral proteins, carbohydrates form special sites on the cell surface that allow cells to recognize each other. This recognition function is very important to cells, as it allows the immune system to differentiate between body cells and foreign cells or tissues. Together, the phospholipids in the bilayer create a sea in which other molecules can float. This sea concept is the basis for the fluid-mosaic model of the plasma membrane. The phospholipids can move sideways within the membrane. At the same time, other components in the membrane, such as proteins, also move among the phospholipids. Because there are different substances in the plasma membrane, a pattern or mosaic is created on the surface. A key property of the plasma membrane is selective permeability, or being semi-permeable, by which a membrane allows some substances to pass through while keeping others out. Now consider a fishnet as an analogy of selective permeability. The net has holes that allow water and other substances in the water to pass through, but not the fish. Depending on the size of the holes in the net, some kinds of fish might pass through while others are caught. This figure illustrates the semi-permeability of the plasma membrane. It shows that some molecules, such as gases, hydrophobic molecules, and small polar molecules can enter and leave the cell through the plasma membrane easily, while large polar molecules and charged molecules cannot. Control of how, when, and how much of these substances enter and leave a cell relies on the structure of the plasma membrane.